Have you ever heard of Merkel Cell Polyomavirus, or MCV for short? This isn't your everyday virus. MCV is a fascinating subject of study due to its association with a rare and aggressive type of skin cancer known as Merkel cell carcinoma. The virus is ubiquitous, found in a significant portion of the general population. But here's the kicker. While many carry this virus, only a small fraction ever develop cancer. Intriguing, isn't it? MCV has a knack for integrating into our DNA, expressing proteins that can cause cancer, and sneakily evading our body's immune responses. Unraveling the mystery of MCV's molecular mechanisms and infectious details is pivotal. It could lead to improved diagnostic methods and the development of effective therapies for Merkel cell carcinoma. It's a common virus, but it has the potential to cause a rare, aggressive type of skin cancer. Let's delve into the molecular mechanisms of MCV. Imagine MCV as a cunning infiltrator. It utilizes glycosaminoglycans and sialylated glycans as attachment receptors to gain entry into cells. But not just any cells. This virus shows a special preference, or tropism, for dermal fibroblasts, endothelial cells, and Merkel cells in the skin. It's like a sneaky intruder who knows exactly which doors to knock on. Once inside, MCV starts to flex its muscle. Its early gene region encodes two major oncoproteins, the large T antigen, also known as LT, and the small t antigen, or ST. Think of these oncoproteins as double agents working inside the cell. The LT antigen binds to and inactivates tumor suppressors like ARB and P53, effectively disarming the cell's security system. This promotes cellular proliferation and genomic instability. It's like giving the keys to the city to a band of unruly invaders. On the other hand, the small t antigen also has its share of mischief. It's capable of transforming rodent cells and disrupting the PI3K/ACT pathway. This contributes to cell survival and motility, essentially paving the way for the virus to take over and establish a stronghold within the cell. But wait, there's more. In most Merkel cell carcinoma cases, the MCV genome is integrated into the host cell's DNA. This integration often leads to truncation or mutation of the LT gene, making the protein even more oncogenic. It's akin to the virus planting its flag in the cell, claiming it as its territory. What's more, MCV has ways to evade the immune system. It encodes proteins like viral miRNA and a viral protein that can downregulate the expression of viral antigens. This helps the virus fly under the radar, evading immune recognition. But that's not all. These oncoproteins are just the beginning of MCV's complex interaction with our cells. The virus has more tricks up its sleeve. After gaining entry into the host cells, MCV doesn't just sit idle. It integrates its own genome into the DNA of the host cell. This integration can often lead to a truncation or mutation of the large T antigen gene, making the protein even more oncogenic or cancer-causing. But that's not all. MCV also has a strategy to stay unnoticed by the host's immune system. It encodes proteins like viral microRNA and a viral protein known as VP. These proteins can downregulate the expression of viral antigens, effectively helping the virus to hide in plain sight, evading immune recognition. So, while the immune system is busy fighting off other threats, MCV is silently integrating into our genome, mutating, and even helping itself to avoid detection. These mechanisms allow MCV to integrate into the host genome and evade our immune system. Now that we understand how MCV operates at the molecular level, let's look at its infectious details. MCV is a common virus, with prevalence rates among the general population estimated to be between 60 to 80%. This means that a significant number of us have been exposed to this virus at some point in our lives. It's thought to be transmitted through respiratory droplets or skin-to-skin contact, making it easy for the virus to spread. However, not everyone who gets infected with MCV will develop Merkel cell carcinoma. Certain risk factors increase the likelihood of this happening. Immunosuppression, chronic sun exposure, and advanced age are known risk factors. Those with weakened immune systems, those who spend a lot of time in the sun, and the elderly are particularly susceptible. As you can see, MCV is widespread and certain factors can increase the likelihood of developing Merkel cell carcinoma. But how does MCV persist, and what are the implications of this persistence? For starters, let's talk about how MCV establishes a persistent infection. It's a bit of a homebody, really. Once MCV sets up shop in your body, it prefers to stick around, particularly in hair follicle stem cells and dermal fibroblasts within the skin. This persistence is not just a random occurrence, it's thought to be a prerequisite for tumor development. Now, on to the implications of this persistence. You might be wondering how this can affect diagnosis and therapy. Well, the presence of MCV DNA and viral oncoproteins in Merkel cell carcinoma tumors can serve as a kind of red flag. These can be used as diagnostic markers, providing a potential early warning system for the disease. But it doesn't stop there. This persistence also plays a significant role in therapy. Current research is like a treasure hunt, exploring the development of immunotherapies and targeted therapies against MCV oncoproteins. The aim, to treat Merkel cell carcinoma more effectively. Think of it this way. If we can understand the enemy, in this case, MCV, we can better strategize on how to defeat it. This includes understanding how it persists, how it transforms normal cells into cancerous ones, and how it evades our immune defenses. In the grand scheme of things, despite MCV being a common virus, its ability to persist, express oncogenic proteins, and dodge immune responses is pivotal in the development of Merkel cell carcinoma. This is a rare but aggressive form of skin cancer. Understanding these infectious details is crucial for improving diagnostics and developing effective therapeutic strategies against Merkel cell carcinoma.